Okay, so then I will start this webinar. Good afternoon, everyone. My name is Jappe de Best. I'm working at the Center of Expertise Bio-Based Economy of Avans University, and today I will be presenting or chairing this session, which is organized by the Grassification project, which is an Interreg 2Cs project. Echo. So today, to give a brief overview, Grassification project wants to valorize roadside cuttings from grass, and of course you can do that in different ways. And we already had a webinar a few weeks ago where we focused our attention on the fibers coming from roadside grass, but today we will have a session which focus on the liquids which can be extracted from the roadside grass. So on the screen you can already see an overview of the different presenters today. And well, let's put it on, excuse me, because I first would like to introduce the first speaker of today, which is Marcela Fernandez de Souza. She is a chemical engineer with a PhD on technology of chemical and biochemical processes, and she is currently the project manager at Kent University. And her research interests are bioprocesses, biomass valorization, and resource recovery. And she has a busy schedule today because she's presenting twice, but today she will start telling you a bit more about using grass juice as a nutrient source for microalgal cultivation. And if you have questions for Marcela, I would like you to type them either in the chat or raise your hand. And then after the presentation, we have time for 1 or 2 questions before we go to the next speaker. So I would like to give the floor to you, Marcela.
Thank you, Jappe, for the nice presentation about me. Yeah, I'll just open my window. Yeah. Do you see it properly?
Yes, we do.
Yes, okay. So yeah, good afternoon, everybody. Thank you for joining this webinar on the uses of grass juice. So we will start with a focus on using the grass juice as a nutrient source for microalgal cultivation. And since I'm the first one speaking in this webinar, I would just like to give you some background information around the grass juice, or the liquid fraction of grass. The terminology varies depending on the person producing the knowledge. So sometimes it's you'll find different terms on this, but let's call it grass juice for simplicity. So why do we want to produce grass juice in the Grassification project? As Jappe introduced, we are looking into valorizing roadside grass clippings, which means that you generate the grass clippings in a very specific area, which is the roadside verge, and then you have to transport it into the facilities that will valorize it. And if you can separate the grass juice from the grass fibers, you can reduce this transportation volume. At least even if you have a first moment where you're going to take all these grass clippings from the verge and transport it into a processing facility, if you can already separate the juice from the fibers in this first one, and then have these 2 streams being taken to other facilities where they will be further processed, you already reduce the volume. And ideally, the grass juice would not go very far, because every time you transport liquid, you have a low density and higher costs. We also want to remove the readily available organic molecules from grass fibers. So when we separate the juice from the fibers would go into further processing for biomaterials and biocomposites, for instance. And there you don't want to have readily available organic molecules that can be consumed by microorganisms and might actually make your product very biodegradable. So this can be also an advantage, that you take these organic molecules with the grass juice, and then the fibers are less prone to contamination. And also, we want to increase value by further refining. So if you just use the grass fibers and dry them, you only have one type of product. If you refine by separating the juice from the fibers, then you can actually have these 2 streams that will be further valorized. So how to produce the grass juice? So Grassification used a screw press. So I have two little videos just to give you an idea, as image can be better than words. So here you see this was a demo day. You can see that the grass is fed into the press, and then you have a screw that just goes to feeding the grass into the equipment, and it's by just a sheer force you have the separation of the liquid from the fibers. And then you can see here you have the fibers coming out of one way and then the liquid coming out of the other part of the equipment. So this is this was the chosen technique. It's what we have been seeing in the market, but there might be other ways to separate as well. We did investigate that. So just a little background on the quality of the grass juice as a nutrient source. In Grassification, we did 2 trials. Oh, I hear myself. Okay, I think it's fine. So we did 2 trials with different mowing heads. So the actual equipment that will take the grass clippings from the roadside verge. One was a flail mower, the second was a circular mower. And we did see some changes in the quality of the grass juice being produced. Probably because the flail was able to produce smaller fibers, so there were more points in which the grass juice could be extracted, and then you have different solubilities, and this impacts the nutrient source. But you can see that the stream is somewhat rich in nitrogen, potassium, and phosphorus, being an interesting nutrient source. So more specifically, now going to the topic that I want to talk about today is using this as a nutrient source for algal growth. So we have a nitrogen to phosphor ratio in the grass juice of 3.2. Well, this was specific for this grass juice. This will vary depending on the type of grass, the season in which you will harvest it, but this specifically was 3.2. The suggested one for algal growth is actually from 9 to 16. But a higher nitrogen to phosphorus ratio is actually not a problem. It just means that possibly, not all the phosphorus will be consumed. Well, not all the nitrogen will be consumed. Maybe phosphorus will be a little bit. Yeah, so we will have a little imbalance, but it's not really a problem. The biggest problem is when you have a higher nitrogen to phosphorus ratio. So this suggests it can be a good medium for algal growth. It's also rich in minerals. So this also suggests that we wouldn't need to give more nutrients to the algae, that the grass juice would already be enough. However, it has a dark color. If you remember from the video, it was quite green and not really translucent. So this can be a problem since algae are photosynthetic organisms and do need light to grow. And it's also rich in sugars, which I put a question mark because this can be good. So some algae can consume these sugars and just boost their growth with these organic carbons. However, this also means that you can have a lot of bacteria, yeasts, and other organisms also growing there and competing with the algae. So this was something to be evaluated. So the chosen algae to use is actually a cyanobacteria. Its scientific name is Arthrospira platensis, but its commercial name is spirulina. You can see in the pictures how it looks in the microscope and also how the products look, because this is already a commercially available algae that's used as a superfood because it's rich in proteins and also has some important minerals and antioxidants and other very healthy molecules. So that's why we chose this, because it's already generally regarded as safe and it would be readily available for market. So the first observation that we had was that grass juice is not suitable undiluted. So as I said, it has a very dark color. If you don't dilute it, the algae just don't grow. So I tested different dilutions, and you can see that you have the control with the mineral medium. So this is the perfect medium for spirulina growth. And I also had different concentrations of the LFGs, the liquid fraction of grass, so the grass juice, together with this medium. So the first assessment was whether to see if the grass juice was toxic. And you can see up to 20% it was fine. The growth was even a little bit higher than the mineral medium, so this was already a very good indication. But we did need to dilute. If I would use 30% already, the algae would not grow, most likely because of the lack of light going in the medium. The second thing that we tested was if we could use the diluted grass juice as the sole nutrient source. So no more adding any mineral medium, just having the grass juice and the algae. So we noticed that you can. However, there was a problem with pH. So specifically for spirulina, they need a more basic pH, so higher than 7. It's usually from 8.5 up. And the grass juice had a very low pH, so it was around 4. So it was necessary to increase this pH. And we tested with 2 different types of chemicals. So the normal base, the sodium hydroxide, this did not yield a good growth. However, using the sodium bicarbonate, which is a very common already component of the spirulina medium, which also gives inorganic carbon, so boosts algal growth. This was a very good way of adjusting the pH. So you can see that the mineral medium and the 5% grass juice with the bicarbonate gave similar growth. So the other experiment was just to see how much grass juice we could feed to the algae. So at the end, 15% was the ideal concentration. And this is because, as you can see in the graph, we tested from 5 to 20. Actually, 20 looks very good. However, sometimes the culture would crash because it's already a very dark medium. So it would depend if the cells were very healthy and depend on the inoculum size. So 15% was one condition that always gave good results. That's why we chose it. And also, if you can look at the picture on the right side of the slide, you'll see that the 5 and 10%, even though they had good growth, the color was a little bit weird. And that's because it was probably lacking nitrogen. So it would produce less of the pigments in order to be able to grow well. So 15% gives good growth, the culture would not crash, and it had a healthy color. And the pigments are an important part of the spirulina commercial value. So we do want to have a condition where you have very healthy cells with this very dark green color. So a few conclusions. So the grass juice was suitable for growing algae. However, specifically for spirulina, we needed to correct the pH. And as a step that's still missing is the quality of biomass still needs to be assessed. So, of course, we had this first assessment on the color, which the cells looked very healthy with the 15% grass juice, but we still don't know how this grass juice is affecting the composition of the algae. So this remains to be assessed. Just some future perspectives, which are actually already ongoing. Since we had these interesting results with using the grass juice as an algae medium, we submitted an operational group called Grass to Algae for the Department of Agriculture and Fishery of Flanders, and it was successful. So this operational group is running since January this year. It's a little bit different from grassification. In grassification, we do propose the roadside verges as a grass source. And in this one, since it's from the agricultural department, we focus more on the edges of farms where you also have grass growing and it's not really useful. The farmers need to mow it. It's a cost and they want to see how to valorize the stream. So we have the grass juice, which you can see in the picture in the middle. It's quite dark. And this is really fresh juice. So for the grassification, all the experiments we performed was in a grass juice that had been frozen. This was just for logistic reasons. We produced a lot of grass juice, then we froze it, and then we started doing the experiments. For this grass to algae, we chose to have different pressing moments so we can work with the fresh juice. And it gives a lot of different properties. So actually now in the grass to algae, we're investigating a first sedimentation step as a pretreatment, and then we would just have this upper layer reaching the nutrients and very translucent. So you can see in the picture that is already quite light. So it might give a very good result. So this is still ongoing, and we hope to have results soon. So that was it. Yeah, if you have questions, you can ask now, or you feel free to contact me directly. And we have the Grassification website. So thank you.
Could you hear me, Marcella, or was I still on mute?
No, yeah, I couldn't hear you, but I was wondering if it was my problem. Sorry. Yeah, no, it was
New
from your side.
microphone, so that was. I will read it. So there are two questions in the chat, and there's one question from Johan Raap. He asked, "Do you see differences in quality between grass harvested in the morning or in the afternoon?"
Oh, that's a good question. We did not evaluate this in grassification. From what I read in literature, I do see a lot of studies pointing to differences between summer and spring and autumn, so seasonal variation. I do not know specifically for morning or afternoon. It can be there are some differences. I know that the grass has, of course, different, so the photosynthesis, the respiration, so this might somehow impact the quality, but maybe not so much, because we did have 2 batches of grass juice coming from grassification, and honestly the differences in nutrients were not that high. So it can be that these daily variations do not impact that much, but yeah, something to be studied.
Okay. And since you said there was no much difference in the juices we had from grassification, which I think was one just before summer and one after, could it also have to do with the timing, or the time the juice was left before it was frozen, or was it frozen directly?
Yeah, it can be. So all these things, that's why in the Grass2Algae, we're really working with the fresh juice, because yeah, in grassification, the juice just stood there for quite some hours before we were able to bring it to the lab, and it was a hot day. So definitely, I will actually show a little bit of storage results in the other presentation, but yeah, definitely this has an impact on the quality of the grass juice.
Okay, thanks. Well, then we'll wait for your second presentation. But first, there's one other question, which is from Rosine de Ruiter. She asks, depending of the location of the grass next to a highway, for example, I imagine that the juice could contain contaminants like heavy metals or other contaminants that can affect the growth of the algae. Did you study that, or has that been studied before?
Yeah, so we are assessing this in grassification, the heavy metal content of the roadside grass clippings. This was also assessed in a previous project from France, also looking into road verges. Since we have now good filters in the cars, we don't really see heavy metal contamination in the roadside verge, because yeah, that's legislation wise, the cars are not supposed to be really releasing heavy metals, so we don't see much contamination. There is one thing that we are investigating, is to see in the exit of the highways, maybe you have a higher heavy metal contamination just because of the braking, and there you can have some heavy metals coming out of the brake pastels. I don't know how to say this in English, but we are looking into it. But this would be a very specific contamination in a very narrow area. But overall, we don't see high heavy metal contamination.
Okay, and do you see any effects from the tires, because they also wear out?
Yeah, that's we really didn't see so the British partners are doing some tests in the microplastics, but yeah, this wasn't a focus on grassification, so we might have some results, but yeah, I don't have anything. But yeah, well, the first results didn't point as a problem, so we're hoping that the grass juice overall is good.
Yeah. Okay, thanks very much, Marcella. Then I would like to go to our second speaker, which is Naomi Rintal. Naomi is Senior Lecturer in Soil Science and Environmental Management at Canterbury Christ Church University. And for the Grassification project, Naomi has focused on quantifying the ecosystem services provided by road verges, and also she looked whether the liquid fraction can be used as an eco-friendly, sustainable biofertilizer. And today Naomi is going to tell something about the development of a road verge liquid fraction biofertilizer products. And it also says preliminary results. So I'm curious. The floor is yours, Naomi.
Brilliant. Thank you. Right, I'll just get this up. Okay, can you see that slides okay?
Yeah, I can see that now.
Brilliant. So, yeah, I'm Naomi Toole. I'm at Canterbury Christchurch University. And so what we're interested in is, once this grass has been collected and pressed, is this really nutrient-rich, dark green liquid that we get at the end of it. And what interests me is, if it's nutrient-rich, there's surely other uses for it and products that we can develop from it. And the reason I've put preliminary results in there is because this is really just the start of research and development onto whether we can produce any sort of biofertilizer product. So I'm going to present the first experiment that we did, and then we're on to the second experiment based on the findings from that. So the first thing that we did with our grass liquid fraction, once we got it, was we just wanted to know whether adding that untreated liquid fraction to tomato plants, whether it could actually improve the plant growth and nutrition. And we compared that to, we wanted to compare it to some other treatments as well. So we used just tap water. We used Tomerite, so an inorganic fertilizer. We used Verve, a plant-based product, it's actually seaweed-based product. And we used the grass liquid fraction, the grass juice as well. To keep it consistent, we made sure that we had the same amount of nitrogen per plant per week. So it was 0.6 grams of nitrogen per plant per week added. And that meant the only thing that we did to any of these was we diluted it accordingly. So they got the same amount of water each time, but with a different amount of fertilizer in each sample. And there were 10 of each pot, 10 tomatoes with each of these 4 treatments. Just on the back of that question about the heavy metals, that was something that that comes up quite a lot, and so that was one of the first things that we wanted to look at. And so we thought, because it's roadside, it could potentially have high levels of heavy metals. As Marcella said, we know it actually doesn't have particularly high heavy metals anyway. And actually, when you look at, one thing I was concerned about, would it be translocated into leaves potentially? And what we found was actually the liquid fraction didn't seem to have high heavy metals, and also the plants didn't seem to take up any more heavy metals. In fact, there was a significantly lower concentration of copper and cadmium in the grass liquid fraction, the leaves that had been treated with the grass liquid fraction, than the other plants. There wasn't a significant difference in chromium, and things like lead and nickel that you'd commonly consider look at for potential road verge pollutants were negligible. So the first thing we know about the liquid fraction is we don't need to particularly worry about heavy metal concentration. It doesn't seem to be a problem. The other thing that commonly comes up is salinity. So we thought that there might be an issue with salinity, in that if we are gritting and salt spreading on roads when it's icy, that could lead to particularly saline soils. It could lead to salt in the vegetation, which then, when it's pressed, it could lead to highly saline liquid fraction. And what we found was it was more saline, but not at levels that we would be particularly concerned about. So we're looking at the averages there of 800. It'd have to be about double that to be even really classed as slightly saline, and it would have to get to more like 4,000, 5,000, 6,000 before it would be classed as highly saline. So the liquid fraction did not cause the soil to become saline. So that's another thing that we don't necessarily need to worry about, although, as we've mentioned, it might depend on the time of year that you're collecting the vegetation if there's a lot of salt spreading. And the other thing was that because, as Marcella said, it was a very low pH liquid, about 4.5, we checked whether that would have an impact on the soil pH, and it didn't. There was no significant difference between the soil pH between those different treatments. So those are some good points that we know, that the things that we don't have to treat the liquid fraction for. That's kind of where we got those good points, and then there were a few things that we didn't necessarily expect to happen that we then had to kind of deal with. So what we really quickly found was, in the plants treated with the liquid fraction, that within a couple of weeks of being treated with that liquid fraction, immediately you could see that the leaf colour was much yellower. So this is just a grading scale of the lower the number, the more yellow the leaves are, so from 1 to 5. And so what we found is actually the liquid fraction leaves showed signs of plant stress within a couple of weeks of being exposed to liquid fraction. That was a significant result. And so we also looked at the nutrient content in those leaves as well. And what we found was there was a significantly lower concentration of nitrogen, phosphorus and potassium in the liquid fraction than those other treatments. And when you think that we added a known concentration of nitrogen, 0.6 grams per plant per week, that means that even though they're getting the same amount of nitrogen into the soil, that's not being taken up to the leaves to the same extent in those plants treated with the liquid fertilizer. When we looked at the growth, we also found that growth was affected above and below ground. So above ground, the height was much lower for the grass liquid fraction. That was also quite clear very early on, and the biomass was lower. And with the root biomass, what we found was because we'd grown those plants for 3 weeks before we transplanted them into larger pots and started adding the liquid fraction, when we took the liquid fraction pots apart, we realised that the root ball that was there was the original pot root ball. It was the same shape as the original pot, and the roots had barely grown since we'd started to add the liquid fraction. So it really negatively affected the root growth. And the other thing that you're going to want with tomato plants is you want tomato fruit, you want fruits off them, you want a large crop, and we found that that was significantly affected. So not only actually there were less ripe tomatoes that came off the liquid fraction plants, but also they were quite a lot smaller, so smaller biomass, smaller diameter as well. So we're getting small and irregular tomatoes. And finally, you definitely, if you're growing tomatoes, don't want plant mortality, and we got quite significant plant mortality only with the liquid fraction. So this is the sort of plant we were getting, very yellow leaves that one died off from week 2, week 5, 6, 8, and we actually, out of those 10 plants, we only had 4 that survived through the whole 16-week experiment. So we know there's some positives, that we don't have to worry about heavy metals, we don't particularly worry about salinity, it didn't affect the soil pH. But we know there's still these negative effects, so we had to look at what caused those and how we could potentially rerun that experiment and deal with those problems. One of them was that we got this film of fungi on the pots, and we got this presence of bacteria. And with the fungi, first of all, it was found to be pathogenic, so that could be partly the cause of plant death, and this was only on those liquid fraction experiments. And we also started to get bacteria, these black spots, the samples kind of rotting. And what we've realised with those was that when we were adding the water and these had these saucers underneath, with every other, all the other 3 treatments, the water was getting taken up by the plant quite readily. With the liquid fraction, the water was sitting there and not going anywhere, so the roots weren't taking up the water and the plants were effectively rotting because it was waterlogged for much of the time. And so those were the first 2 issues, that we had this fungi, we had this waterlogging and potential bacteria. The other thing that we started to get was root damage. So we know there was issues with the cortex of the root. We know that they were a different structure. And there's 2 potential issues with the root damage. We think that possibly that's down to, although the pH of the liquid fraction was very low and didn't affect the soils, it might have still affected the plant roots if the roots are taking up a very low pH liquid. So we know that low pH might affect things like the nutrient availability, nutrient uptake, and the root health itself. So that could have been due to pH. And it could also have been due to the fact that lots of these species that you would find in a road verge will contain allelopathic chemicals. And when you press these grasses and you get the liquid out of them, you effectively can have some of these chemicals in the liquid as well that will affect the plants that you grow in them. And we know that some of these negative effects of allelopathic chemicals include things like an increase in cell permeability, leading to slow growth and death. We saw that. An inhibition of nutrient uptake, we saw that. We saw that there was less nitrogen, phosphorus, potassium in the leaves, for example. An inhibition of root elongation, cell division, cell ultrastructure. So we know that there's potential that that's what caused the root damage as well. And the final things we saw was that it was very high sugar content. That might be why we got such extreme fungal growth only on the liquid fraction experiments. And also we could see the kind of rainbow haze, the sign that there's some sort of fuel in there. And we don't think that's fuel that's been taken up by the plant. It's fuel that's been sat on the plant parts, on the leaves, etc., that when it's been squashed has gone into the liquid. And so these are all issues that we then had to think, right, what now? This experiment hasn't worked, and the plants it's not improved plant nutrition, it's had the opposite effect, but it's the first stage of research on this. And so what we decided is actually the next step is, now that we've worked out some treatments that we need to do, we need to rerun the experiment, treat the liquid fraction, and see whether we can then get a plant which responds positively to this liquid fraction. So we're now just setting up our second experiment on treated liquid fraction. And we're going to look at the controls, the tap water and the other fertilizers that we used before, and compare that not just to the untreated liquid fertilizer, but to liquid fertilizer which is treated by fermenting first to try and remove some of the sugars, particularly because grasses tend to have a lot of soluble sugars rather than the sort of complex sugars. So we think that could be a big issue. We're going to pasteurize it. So by heating it, we might remove some of those fungi and bacteria species that had a negative impact. We can pH adjust the liquid fraction to bring it closer to a neutral pH. And finally, we can add bacteria. So we have some harmful microorganisms, but we can add some potentially beneficial microorganisms. So bacteria that will effectively eat up any diesel fuel in those samples and try and remove that from the samples. So this is the next stage of our experimentation with this product. It's not necessarily our final stage, but what we hope to do is to keep experimenting with it and tweaking that liquid fraction and until we get to a point where we know how we can treat it, that we have a product that is sustainable. It can be produced, for example, in the UK and in our county in Kent. It can be from our road verges in Kent. It can be processed in Kent, and it could be sold to gardeners in Kent. And so it's a really nice sort of low carbon sustainable product by the end of it. It's still ongoing, so hopefully experiment 2 is more successful in terms of the plant nutrition. Thank you.
Thank you very much, Naomi, for your presentation. Well, again, I'm looking at the chat. I see some questions which I guess are already answered, but I also see a question. Did you check for other ions like chloride and sodium?
Yeah, we did check for several different things. There was nothing that we found that we were particularly concerned about in there in terms of something that would be too sort of toxic or dangerously high. There were some other things with, for example, sodium was quite high. So there was, which we kind of would have expected because the salt's quite high. So there's some other things that, again, we can potentially treat and get back down to a level that we'd want.
Yeah. Okay, and I see another question about, did you test what kind of fungi or bacteria were present?
Yes, we did. It was Aspergillus species. So it's it was a pathogenic species, which we think is possibly why we're getting plant death there.
Yeah.
We haven't tested the bacteria yet. We don't know too much about the bacteria, but we know there's probably bacteria present that may also be having an impact.
Okay. And then I see something about, could the organic content, sugars, proteins not be too high? But I guess that's what you're going to basically look into by trying to remove them.
We're going to try and remove the sugars. Yeah, we know that there was the organic content's fairly high. The soil organic content by the end of that 16 weeks was significantly higher than the other treatments. So we know that the organic content is quite high. But I think the most concerning bit is probably the sugars for us. That's probably causing that the fungi to be produced. So that's something we have to deal with.
Yeah. Did you ever find, I'm curious because you see that Marcella just told about the algae which grow perfectly fine with the same liquids. Any explanation for that or any idea why that would be? Maybe Marcella does have an idea.
Sorry, for what, sorry?
Yeah, with the algae, you see they perfectly grow on the liquid, whereas for the plants they don't grow. So then for me the question would be, what's the difference in this case? You probably have thought about that before. I see Marcella laughing, but any idea?
No, yeah, so I will also talk about a bit in my presentation. So maybe we take it again in the Q&A if there are still some remaining points to be answered, but I will cover it a little bit.
Then, well, I'm looking forward to the presentation. So thanks, Naomi. And before cutting more grass in front of your feet, I'm not sure if that's an English expression, but I just translated it straightly from Dutch. So I would like to go to thank you, Naomi, to the third speaker, which, well, I'm not going to introduce you again, Marcelo, maybe just adding that you're also the project leader of Grassification. So that's a role I didn't mention before. But for your second presentation, you're going to focus on the anaerobic digestion of grass juice for energy production. And apparently you also touched some of the topics that I previously already mentioned. So I would like to give the floor again to you, Marcella.
Thank you, Jaap. Yeah, I did promise a lot of answers. I do hope I will deliver them. If not, I'm happy to discuss later on. So yeah. Here we go. So yes, what happened? Okay. So yeah, the topic is anaerobic digestion of grass juice for energy production, but you will see that I'm going to also cover a little bit around storage, and also in the future perspectives, we do come back for the whole fertilizer thing. So this was developed together with Inagro, and the main researcher behind it's Audrey Miseres, but she couldn't be here today, so I will be presenting on her behalf. That's why I have two presentations. So first, how to produce energy from grass juice. We chose the anaerobic digestion technology, which is the perfect technology once you have an organic stream and want to get the energy out of it. So it's usually used for animal slurry, energy crops, and food waste. And now we are proposing to use the grass juice as also a feedstock for this technology. So once you feed this organic rich stream to the anaerobic digester, there you will have a population of microorganisms that will be able to break down this organic molecules into biogas, which is our energy carrier, and also a nutrient-rich digestate, which can be used as fertilizer. And that's how I will also link it to Naomi's presentation. So from the biogas, you can feed it to a CHP unit and have electricity and heat produced. Just a little bit of advantages and disadvantages of using the grass juice as a feedstock for biogas production. So being a liquid stream, it's composed of mostly soluble components, so it should have a high digestibility. Contrary to, for instance, when you give energy crops, in which you really have to have a microorganism population that will be able to hydrolyze the solid parts of these crops and bring them to soluble components for the other organisms to use it for biogas production. So in the grass juice, most of the components are already there available for biogas production. However, it is a highly diluted stream. Of course, it's liquid, so the energy density, it's not expected to be very high. And it's also highly reactive, which means we already discussed this a little bit. The moment you press the grass and you have the juice, it starts to decompose. A lot of things are happening there. You see gas production. So we were wondering how is the main nutrients are being transformed, and if there is any easy storage possibility that we could use. So let's begin with the storage. Inagro conducted an experiment in which they tested the fresh grass, so 1 day after mowing. Also stored this grass juice as in the fridge, so at 4 degrees Celsius for 1 month and 2 months. And also they left it at what they called room temperature, but it was actually a controlled incubator at 21 degrees Celsius with a 60% relative humidity, also for 1 month and 2 months. And they did some characterization of these grass juice after the storage time, and I'm going to show some to indicate what was happening. So first was caldol nitrogen, which is not total nitrogen, so caldol doesn't account for nitrate, for instance. That's why it makes sense. We are seeing a decrease in the caldol nitrogen with time, and it really doesn't have a lot of difference between the fridge and the room temperature. Yes, room temperature is a little bit faster, but at the end, even at 1 month in the fridge, the caldol nitrogen was going down. So this means that the nitrogen is being mineralized into nitrate, because caldol doesn't measure nitrate, but it can also just be nitrification and denitrification, so it can be just N2 gas also being released. We do see gas production when you leave the grass juice in a closed container. They also characterize ammonia. And it's interesting that in the first month in the fridge, we actually see the ammonia going high. which makes sense with the mineralization of the nitrogen. So the nitrogen in the grass juice is mainly in the form of organic molecules. So you have chlorophyll, which is rich in nitrogen, and also proteins, amino acids. And so the organisms that are already present in the grass juice, they're clearly mineralizing this into ammonia and probably nitrate, and then nitrogen gas. So in the fridge, since this process is a little bit slower than in the room temperature, we actually were able to see what seems like an in-between step, so from the amino acids to ammonia, while in the room temperature, actually it remained the same, and then it went down after 2 months. So yeah, there's a downward tendency at the end, because probably it's going to nitrate and nitrogen gas. They also analyzed the BOD and COD. Interestingly, again, in the fridge, we have a little increase in the first month, but the overall tendency is going down. So you see that indeed there's a lot of microbial activity just in the grass juice. So without inoculating anything, there are a lot of reactions going on there that change the composition of the grass juice. So finally, they went for the biogas potential with the 3 streams that they had. So the fresh one, the fridge one, and the room temperature one. So you see that the fresh grass juice gives the much higher biogas potential. So indeed, it's complicated to store this stream, at least with the techniques that were evaluated here. So room temperature, definitely no, you really lose more than half of the biogas potential. The refrigerating it is a little bit better, but still you lose a lot of biogas potential. So interestingly, the methane content didn't change, so it was always around 60%, but the overall biogas production was really affected. So storage was not possible. If you really want it for energy production, you just lose a lot if you don't use it immediately. So there are some possibilities. We might be able to ensile the grass before pressing, and then you have afterwards the pressing the minute you need to use it for biogas production. We didn't test this, but I am aware that there are some people using the ensiled grass juice for anaerobic digestion, and it has a good energy potential. So this can be a way that you just store the grass, and every time you need to feed the digester, you press the amount of grass that you need to generate the grass juice. It can also be seen as a seasonal input for anaerobic digestion, for cold digestion. So imagine you already have a digester. It's operating with other feedstocks, and the moment that the grass verge is mowed, you could press it and just have this as an additional input for that moment to boost your energy production. A third option would be to just have monodigestion, so you would only digest the grass juice and use as an organic fertilizer, but again, only during summer and autumn you would have this availability. And we just wanted to test if this third option will be possible. So we have the future perspective using the digestate from the grass juice as fertilizer. So here my presentation links with Naomi, and I hope to answer some questions. So at UGent, we worked very closely with Canterbury Christ Church University, and we did a soil incubation test where you just have the soil with its own microbiota, and you add the stream that you want to test as a fertilizer to see how the mineralization occurs. So we have just the soil sample with only water. So you can see that it does increase a little bit the nitrate content, which means that the organic nitrogen is being mineralized into nitrate. What we would like to see is this, that really goes from a low nitrate content to a high nitrate content. So this is an ammonium-based mineral fertilizer that we use as a control, a positive control. So this is what we would like to see. And this is what we saw with the grass juice. It was actually even lower than the soil, which can explain in part some of the results that Naomi presented. It's because the nitrogen is it's an organic form, so it should go, it's complex to go to nitrate, it needs to go to several steps, and you do have a lot of sugars there. So yeah, I'm going to already show you. So the total nitrogen was 3 grams per liter. However, the mineral nitrogen, so nitrate plus ammonia, was only 0.4 grams per liter. So the organisms really need to take the nitrogen from an organic, a complex form, to change it into a mineral form. So this already demands more of the microbial community in the soil, and you have a very high content of sugars. So we saw 80 grams per liter of sugars, which will unbalance your whole soil microbiota, will make other things grow, like Naomi suggested, bacteria, fungi, which actually start to uptake the nitrogen. Instead of leaving it available for mineralization, they just uptake the nitrogen with the sugars to produce microbial biomass, which is not what you want in a healthy soil. So what we are hoping to have is that the anaerobic digestion system will help to mineralize this nitrogen. So it will take from the amino acids, chlorophyll, and actually make it into ammonia, which is a more readily available nitrogen that the soil will be able to mineralize into nitrate for plant uptake. And also it will transform all the sugars into biogas and leave it with a much lower organic carbon concentration that will hopefully leave the soil healthy. So what we are doing now is a continuous digestion of grass juice, because what Inagro did was just a biogas potential, which you add a very little amount of grass juice to a very high amount of inoculum. And now we're just doing it properly, like you would do if you're applying the technology. So every time we feed a little grass juice into the digester until you replenish the whole volume 3 times until you have the proper grass juice digestate for use as a fertilizer. And we will be testing it with flowers because they're easier to grow than tomatoes. The tomato experiment was really, had a lot of work input. So we're testing with flowers, because we're also hoping that perhaps a municipality could use this, mow its own verges, press the grass, have the grass juice digested for energy production for its own use, and then perhaps use the digestate as a flower fertilizer for the city hall or other public buildings. So this is currently being done. You can see in the picture our flowers are still very tiny, so we will hope to have results soon. So again, if you have any questions, just please ask away or contact me later on. Thank you.
Thank you very much, Marcella. And of course, there are some questions. So I'll just start on the top. As the question is, why grass liquid for methanization? Why not the total grass as an input and dewater afterwards?
So the whole idea of separating the grass fibers from the grass juice was to valorize the grass fibers into biomaterials and biocomposites. We are testing in Grassification a small, I would say anaerobic digestion of grass as a whole, and we want to see if the digested fibers would be useful as a material as well. If they are, this can be also a proper way to have digestate and also use it as fertilizer. But our first results are that the fibers really lose their integrity when going through anaerobic digestion. So we are doubting if they will still have just as good properties as a fresh grass fiber.
Okay, and I see a question from, I think his first name is Andreas. He has a question about what's the carbon-nitrogen ratio, and I think it refers to the experiments that you did with the soil.
Oh yeah, that's I think it was around 10, but yeah, I don't have this by heart, but there are reports in the website that will tell you this, but I think it was around 10.
Okay, and I was wondering, because you mentioned that the ensiled grass could also be used as an input. Do you know, as a comparison, in the end, the methane, amount of methane you produce compared to fresh liquids that you tested, or that?
I don't have it. We wanted to test, but at the end we didn't have time, and Yeah, it was just already a huge project. We had so many things to test, but yeah, it's something that we wanted to test. It should be fine. Yeah, during siling you have lactic acid production, but it should not be a problem for the digester since you have a lot of acids being produced anyway there. And I am aware of other people already using this ensiled grass juice as a feed for an Arabic digester, but in co-digestion. So I don't know if it would work for monodigestion, but I have to say I did not find it in literature. I just know from talking with stakeholders that some people are using, but I don't have numbers.
Okay, thanks. And then the last question, well, I guess the whole grassification is focused on cascading in optimal use of all the different fractions. And then Johan Raab mentioned, okay, because you have a high sugar content, would that be interesting for PHA production?
Yes. So this is something actually we have been talking, right, Jappe?
Yeah.
And indeed, so there are so many possibilities with the grass juice, and we are so interested in the stream. It was a very tiny activity in the grassification project. If you see the whole project, it was a very small activity that we had just to see first possibilities of the stream. But yes, we are very interested in further valorizing it and trying different possibilities because yeah, you see with the direct use as fertilizer, it was horrible. But the first experiments with microalgae was immediately positive. So you do have I think it's just a matter of finding the correct process for this stream. And you will see in the other presentations as well, we do already have some leads on other possibilities.
Okay. Yeah, then I'm not going to ask yet what the biggest challenge will be, because then again, maybe I will say something which will be presented later, but I can always ask at the end of all presentations. So thanks again, Marcella, for your presentation. And then I would like to go to our next speaker, which is David de Ruiter from Inagro. And David has a master in biology and a PhD in bioscience engineering. And since 3 years, he is working at Inagro at the Insect Research Center, the IRC, as a full-time researcher, figuring out how to efficiently produce high quality and tasteful insects, I hope. So the floor is yours, David.
Thank you very much for the introduction. Let me share my screen. Put it on big screen. Okay, I hope that this worked.
Yeah, it's visible now.
Perfect. I want to talk a little bit about increasing the sustainability of insect protein production with the use of grass juice. We have talked already quite a bit about it, so I will go quickly on it. The roadside vergers produce about 130,000 metric tons of grass each year here in Flanders. As we have seen, we may be able to use it for anaerobic digestion, composting, grazing, but in this part I will talk a bit more about the use of it in animal feed. You have already seen the clips of how it's harvested and how it is pressed. So we will use also the liquid as animal feed, not for cows or for pigs, but for insects, and more specifically the black-soldier fly and the black-soldier fly larvae. I do not know how well you are aware of insects, but this is on top. You can see the adult fly. The adult fly will lay eggs. The eggs hatch in about 4 days into little larvae, and those little larvae 2 weeks later are ready to be harvested, and they are normally packed with fats, with proteins, are very nutritious to feed, for example, in the aquaculture or to chickens, pigs, and so on. They are not really used at the moment as human food. But when we want to use the grass juice as feed for the black-soldier fly, there are some good things and bad things on it. The good thing is that the crude protein concentration is quite close to what we need for black-soldier flies. Optimally, we have 15% in grass juice, and at least in our sample of the grass juice, we had 11%. However, the main problem was that the moisture content of the grass juice is far too high, 92.3%. It's really a liquid. Oh, sorry. It's really a liquid. So we should not, we can't use that directly. We have tried drying the grass juice via evaporation, but this really didn't work because it turns like into a kind of syrup, a sticky syrup, in which also the larvae just kind of stick and died. So this is not an option. The next option is then to use the grass juice in combination with a dry feed. And we use 3 feeds, spelt husks, wheat bran, and chicken feed. And those 3 feeds also have a different nutritional value, with chicken feed being very highly nutritious, wheat bran is intermediate, and spelt husk almost having no nutrition at all for the black soldier flies. We have tested this we have added water or the grass juice to the different feeds. So we had 6 different diets. We used crate sizes of 60 by 40 centimeter. This is standard in the industry. We added 10 kilograms of wet feed to it. So the wet feed is the combination of the chicken feed, wheat bran, or spelt husks and the grass juice. And we added 20,000 larvae to the box. At 27 degrees and 60% relative humidity, they were able to grow well on it, and as we will see later on. By the end of the experiment, 2 weeks later approximately, we determined the total yield from each box. We also determined the feed efficiency of the total feed, but we also looked at the nitrogen retention efficiency. Now, the first results. On the y-axis, you see the average weight of the larvae, and on the x-axis, you see the different feeds, with CF as chicken feed, the WB as the wheat bran, and the SH as the spelt husks, and with GY being the grass juice and B just water. What we see is actually that, as expected, highly nutritious feed grow better. The chicken feed grows better than wheat bran, and this in turn grows better than spelt husks. But they also grow better with grass juice. Sorry, that again. So this is with grass juice, this is without grass juice. So this means that actually the grass juice does have a significant effect on the growth of the insect, and a significant positive effect. With actually more or less equal positive effect throughout the different experiments or tests. If we look at the feed conversion ratio, we see that the best conversion can be seen again with the chicken feed, the highly nutritional feed, and the worst feed conversion ratio with the worst feed. Just to make a comparison, this is approximately the feed conversion ratio for chickens in general. So we can see actually that the black soldier flies can go below that and can be very efficient with their feed. But very interestingly to see is that with the poorly nutritious feeds, the spelt husks and the wheat bran, we can actually significantly improve our feed conversion ratio. When we then finally look at the nitrogen retention, so how much nitrogen is taken up, we see that actually on the chicken feed, on the left, they're doing very well. On our wheat bran and spelt husks, they're not doing very well. It's better with the grass juice, but then overall, it's actually doesn't perform very well, with only a maximum of about 45% of the nitrogen retained. And finally, we can see that we look at the biomass reduction. The chicken feed is again our control. With the wheat bran, we're getting very close with the spelt husks, the poorest of the feeds we assessed. It remains very high. The reduction is not that good, unfortunately. When we do then the economic analysis to assess the potential of the grass juice, let's assume that we have 1 metric ton of fresh grass clippings. It would produce between 300 and 400 kilograms of grass juice, depending on the season and how much, for example, it rained. We would need to add 125 kilograms of chicken feed to produce 132 kilograms of larvae, which is very good and would be about a 10% savings on the chicken feed. There are some problems, of course, with, as mentioned before, seasonal availability, seasonal variability, because we now assessed it only on a single harvest. We do not know, for example, if there is a difference if you would harvest it in spring or in autumn. The heavy metal pollution is still on here, although we already heard that from the samples used, it was not a problem in our case, but yeah, it might be a problem in other areas. So to jump to the conclusion, does grass have a potential? Yes, it has a potential if it's properly combined. However, it can only be it should be combined with low nutritious feeds, because then it gives a really good boost to that feed. If you already have a very good feed, it's not going to do much. Thank you very much.
Okay, thank you very much, David, for your presentation. I see a question from Federico. He asks you, how do the black soldier larvae deal with cellulose? Would it be possible to feed them on the solid grass cake instead of the juice?
No, well, no. We did a small test with it. It was not the main focus of this project for us. But the problem is the black soldier fly larvae does not have any, doesn't have a real mouth. It can't chew. You have to see it like it's sucking its food through a straw. And it has real issues with fibers, cellulose, anything like that. So we would have to really cut the grass very fine, maybe even pre-digest it or something, maybe then, but raw grass does not work, unfortunately.
Okay, and if you look, because we just had the presentation from Marcella looking at digestion and then of looking at the juice after digestion, could that be an input for the black soldier flies? Did you look at that?
We didn't look at that. It may be a possibility, but the problem is that we actually like the sugars in the grass. The black soldier flies actually like sugars, and it should be even higher, even much higher, not a problem at all. So when it goes to really anaerobic digestion and all the sugars are gone, that's less good. That's good for us.
Okay. And maybe another question, because now you looked at the black soldier flies, I can imagine that there's lots, well, not imagine, there are lots of other insects that probably also can be used for animal feed. Do you think there would be others more interesting to take a look at?
We did some experiments also with the mealworm, as you can see behind me. But the problem is that it is too liquid also for that one. So they do need some kind of moisture source, mostly used now for mealworms as carrots, for example. Mm-hmm. But the problem we had now was the drying of the liquid. And because the liquid was drying into like a honey syrup almost, where insects just got kind of sticking, it was really not very usable for any other insect at the moment. Maybe housefly, but we don't have any population of that one here. But anyway, certainly stay within the fly realm to use them, because those are the only ones that can really use those wet, rotting side streams.
Side streams.
Liquids.
Yeah. Okay, thank you very much. No, I don't think there are any other questions at this moment, so thank you very much for your presentation. And then I would like to go to our next speaker, which is James Gaffey. Is he present already? Yeah, you are. Good afternoon. So James is a principal investigator and co-director of the Circular Bioeconomy Research Group at the Münster Technology University. and he has worked in the field of advanced biorefining since 2009 already, and currently he is involved in several European projects and initiatives on biorefinery. Mention a few: Bio for Africa, COPIT, and Agro Bridges, for instance. And today, James is going to tell more about the analysis of grass biorefinery products from Irish pasturelands. So the floor is yours, James.
Thanks very much for the introduction. So the project I'm going to talk about is a project called Biorefinery Glass, which was an EIP Agri project coordinated by Munster Technological University, where I'm based. And it was an initiative trying to demonstrate grass biorefinery with Irish farmers, particularly dairy farmers. So we had a number of co-ops involved. We had Carberry Group as one of our partners, who are a dairy co-op, where we demonstrated the technology. We had Barry Rowe Co-op, who are a supplier of pig feed, who helped us to analyze some of the products we were producing. We had University College Dublin, who are one of our research partners, and we had Grassa, who are a technology developer from the Netherlands. So just to talk a little bit about the process, and maybe to give a bit of kind of contextual information from an Irish point of view. Ireland is a country where we have a large agri-food sector, but it's a sector where you have a kind of disproportionate amount of emissions. I think New Zealand is the only comparative country in the OECD with the amount of emissions that come from the agriculture sector. It's currently Ireland's biggest sectoral contributor to greenhouse gas emissions at about 35%. So that's a major challenge. Even though there's lots of good that agriculture does, there is pressure on to reduce emissions. On the other side of it, we're also a very dependent country, more than a lot of our EU counterparts, in terms of imports of different types of animal feed. So I suppose the objectives of the project were to try and demonstrate a biorefinery approach for resource efficiency, potentially having the opportunity to reduce emissions, but also to look at how we could displace imports of, for example, soybean meal, and also creating a diversification opportunity for farmers. So this is the unit. It's a mobile biorefinery demonstration facility. It's about 2 tons per hour processing capacity, and it's been developed by Grassa. And it's something that we demonstrated on 5 farms in Ireland. And the main objective was, on one hand, to try and create enough volume of products so that we could test the products and the business model. And on the other side, to try and give farmers some familiarity around the process and involve them in the management of the facility on the farm. So essentially, it's a fresh grass input system. We produce a pressed juice and a pressed cake. The pressed cake is ensiled and it's trialed as a ruminant feed within the project. And then from the juice fraction, we extract the protein concentrate, and we're trialing that as a monogastric feed. And we're left with a whey once we extract those 2 fractions. And from the whey, we analyzed a small amount of this for the extraction of sugars, which are in the form of fructooligosaccharides, which was the target sugars we were looking at, which could be an additive in feed additive and food applications potentially as well. And the residual stream that's left could be used as a bioenergy product or for the production of biofertilizers. So I'll just go through quickly some of the different trials that we conducted based on those products. We did the trials themselves during 2019, and in the 18 months afterwards, we conducted a variety of different trials. So looking first at the press cake, which we trialed as a replacement for silage, which would be the typical winter feed for Irish cows, and in this case, dairy cows. So just to look at the kind of comparative between, we'd say, grass silage and then the press cake silage that was coming off the machine, you can see that the main thing is that you have significantly lower crude protein in the press cake than in the silage. And this is something you would expect because you're pressing out between about 35 and 50% of the juice, depending on the operating conditions. You're pressing out, about 35 to 50% of the protein that's in the grass. You also have a much higher dry matter content in the silage in the press cake compared with the silage as well. So we conducted trials during the early part of 2020 through our colleagues in University College Dublin with 30 autumn calving cows. It was a 77-day trial. And in this trial, we had 2/3 of silage in the control diet being replaced by grass silage in the trial diet. So essentially, you had the same amount of concentrates added in on both diets. The only difference between the trial and control is that in the control, you had 100% silage. In the trial diet, you had 66% pressed cake with 33% grass silage. In the table, you have GS, which is grass silage, and PC, which is pressed cake. Even though the protein concentration was much lower in the pressed cake diet compared with the control diet, you had no significant difference in milk yield or quality in the trial when we compared the two different samples. What we did have was, even though we had the overall nitrogen being much lower in the pressed cake diet compared with the silage diet, you had much of that being converted into milk. You can see in the table that between the grass silage and the pressed cake, it's almost the same. You had a much lower amount of nitrogen, which was ending up in the feces and in the urine, and an overall significant increase in nitrogen use efficiency in the pressed cake diet compared with the grass silage diet. That was the in vivo trial. We also did some in vitro work using the RusiTech approach to model the influence of replacing grass silage with pressed cake in rumen methane production. And we found at the 66% replacement rate that we had about a 15% reduction in methane production in the pressed cake diet compared with the grass silage diet. So overall, it was quite a positive finding. And for Irish farmers, it's important for them to know that they are not going to impact on, for example, milk production by trying to shift to this new biorefinery integration scenario. And then it provides a good platform to start to look at the juice fraction and what you could potentially produce from the juice fraction. So one of the things that we looked at then was the next product was around extraction of the remaining protein that was contained in the juice fraction, separating this and trialing it as an alternative to soybean meal and other ingredients in pig feed rations. So you can look at the bottom of this table. This compares some of the grass protein concentrates with some on the market feed ingredients, particularly soybean meal, which is the main one. In the bottom one here, grass protein concentrate is the dried material. It's about 90% dry matter. You have about a 34% crude protein concentrate, which is a little bit lower, obviously, than soybean meal, but still in the same kind of spectrum with some of the other feeds. Grass protein juice, this is the wet fraction, which is extracted, was much lower in crude protein. So we focus more on this dry protein concentrate as an alternative to soybean meal. And we conducted pig feed trials on a commercial farm at the start of 2021. We conducted this with 55 weaner pigs over a 31-day period. Between the grass protein diet and the traditional diet, which would have been normally used for pigs, weaner pigs at that stage on that particular farm, we were able to replace about 30% of the soybean, 25% barley, and 8% of wheat in the control diet with grass protein concentrate in the trial diet. So what we found in the trial, and it was a fairly high level scoping trial, it wasn't as detailed as the press cake trial. We found that it took a few days for the trial diet pigs, that batch to adapt onto this new grass protein concentrate feed. But once they did, we found that the daily intake started increasing week on week. And by the time we got to the end of the trial, the daily intake was higher in the case of the pigs on the protein concentrate compared with the traditional diet. We also noticed that average daily weight gain started to increase week on week as well, and had reached about 10% higher than the control diet by the time we got to the end of the period. There was no difference in feed conversion efficiency. Animal health seemed to be okay, and the main difference was around the excrement, which is green in the treatment diet compared to normal color in trial diet. We didn't do any carcass assessments. Again, it was a fairly high level scoping, would need a more detailed study to investigate it further, but overall seems quite positive based on our findings so far. One of the things that we did, which I think was quite novel, was to take a portion of the juice fraction. Basically, the press cake, the protein concentrate, and the juice are all produced in that mobile refinery system. We took a fraction of the whey, which is the remaining material once you extract the press cake and once you extract the protein concentrate. And we conducted some lab scale analysis to look at the extraction of fructooligosaccharides from this whey, and we were able to extract about 10 grams of FOS for every 1 liter of grass whey. And FOS has the potential to be used as a prebiotic to promote the growth of good bacteria in animals, potentially humans as well, and is potentially a high value ingredient which could be extracted from grass. We did some cell culture work to understand the activity that using FOS in relation to human and animal bacterial cells could have. We used this and we did some analysis with some control prebiotics. We conducted one which was an on-the-market fructooligosaccharides, which was supplied to us by a company. We did another with inulin, which is extracted from chicory and which is kind of the gold standard for fructooligosaccharides. And overall, we found that there was more or less comparable performance in activities between the grass-based FOS and the on-the-market FOS, which was an interesting finding. And again, this is at quite a low TRL level, but still is an interesting finding and a novel finding. So these 3 pieces of work we're currently working on to develop some publications around, which should be available in the next few months. To try and complement this and try to create the kind of zero waste approach, we also did some analysis on the different streams that are coming off the processes in their potential use for a bioenergy source. And essentially, we looked at the different whey streams, one which was whey coming straight off the machine, one which was whey, which we call defos whey, once the FOS has been extracted. And we also looked at press cake as well. And basically, the reason for this is, and it wasn't supposed to be a big part at the proposal phase to look at bioenergy, and we did look at it at quite a high level, but really we have some energy requirements in the process, both for the separation of the protein. Also, if we want to dry that protein, there's a significant energy requirement. So we wanted to look at the integration of the system with potentially an anaerobic digestion system as well. Another benefit of this, I think, is that at the moment, anaerobic digestion is being proposed as a diversification opportunity for Irish grassland farmers to become more sustainable, but it's the economics of it aren't very sustainable at the moment. So our approach would be to think about trying to link in the biorefinery approach with a digestion process which uses the residual streams of the biorefinery, so that we can achieve a more sustainable, economically more sustainable model. So we looked at the DFOS whey, and this was conducted by Salignus Analytical, who are a biogas company here in Ireland. They found that overall, while the DFOS whey was it was a little bit lower. You can see this on the fresh weight figures at the right-hand side of the table than would say unrefined silage. We still found that it was quite a reasonably good level in terms of having potential of as a feedstock for anaerobic digestion. It also digested in the space of a few days and could be digested within potentially 24 hours as well. So in terms of technically being able to implement this, it doesn't suffer from significant challenges or pre-treatment issues or things like this. So we found that the press cake was more comparable as a feedstock for more comparable in terms of fresh weight biomethane potential than compared with unrefined silage. But overall, we thought on the whey side, which was more aligned with the system that we're looking at, there's still some potential for integration with anaerobic digestion, and we built this into our economic model as well. So just this is the final slide, just on the economic analysis of the system. We carried out quite a detailed economic analysis of the different pathways. We conducted an analysis of 4 different economic scenarios. One of these focuses on a two-product biorefinery, which is essentially the protein concentrate and the press cake. And in this case, we looked at a system which was integrated with anaerobic digestion and a separate system in which the energy was supplied by the grid. So, and then in a separate scenario, we looked at a three-product biorefinery scenario, which is the press cake, the protein concentrate, and the fructooligosaccharides. And again, we looked at this in two phases, one where it was integrated with anaerobic digestion, and in another situation where we have a gas boiler and we have electricity coming from the grid. So we found that the production of the third product, the fructooligosaccharides, it was essential to the economic viability of the model, that unless we included that third product, it just wasn't working economically. The most profitable scenarios integrated the fructooligosaccharides with the co-production of anaerobic digestion, so that three-product scenario with biogas production as well. We found that biogas with biorefinery appears to be complementary, but then it requires further investigation on scaling up of, we'll say, the fructooligosaccharides process and the anaerobic digestion process, looking at the whey. And in terms of the key factors which affect the profitability, the main key factor we found was the cost of the feedstock, and then other critical factors were the prices of the products on sale and also the cost of energy. The capital investment was a less critical factor in the cash flow analysis. So overall, quite positive outcomes from the different products. We're currently working on a number of publications around these, and we have a number of follow-on projects as well in Ireland and internationally where we're looking to develop this further as well. This is our website, and thanks for giving me the opportunity to present.
Thank you very much, James, for an interesting presentation. When I look at the questions in the chat, I see one from Marcella, and she's asking if, based on the potentially available volume of grass juice in Ireland, how much of the soy meal could you possibly substitute? Do you have an idea on that?
Yeah, well, what we've come up with a figure of at least 3 million tons of protein that we could produce if we were to only focus on the winter availability of grass, because we don't think it's realistic. Farmers in Ireland like to have their cows grazing during the early months of the year. They don't want to have that intensive system where cows are kept indoors all the time. So we don't think that is going to happen. So we focused only on the grass silage that's available. And we think that if we were to replicate this to about 75% of available land, it could create 3 million tons of additional feed. How much of that could be replacing soybean meal isn't clear, because we do also include soybean meal in, for example, rations for cows as well. So it's added in. It's not just targeting pig diets and chicken diets. It's also an additional ingredient in cattle diets. So I can't really answer that question except to say that we have said we've come up with a figure of around 3 million tons, which could be created in this way.
Okay. Which seems very substantial, I would say. There's also a question from Rosina. She asked if the winning or extraction of FOS from grass is economically more interesting when compared to commercially available FOS. I think.
Well, we've analyzed our own system economically. We haven't done an analysis of, we'll say, the comparative process, and we don't really know the economics of the system. What I would say is that it's quite a small volume product, but has a much higher value than some of the other products that we're producing in the system. So it adds a lot of value. I don't know enough about the inulin process to give an answer on that, and you know, I wouldn't be able to comment on that.
Okay. Well, I was wondering, because you showed the composition of the different amino acids from the grass protein concentrate, and well, they all are in the same range as the other feeds, except for cysteine, which was considerably lower. Did you look at whether that would be have an effect at all?
No, I think we probably took, I think lysine is a bit lower as well. I think we probably just took that into account really in setting up the diets. We would have started off with the idea that we were going to replace soybean meal in its totality, and we couldn't really do that. We had to do a lot more balancing. So that's why we ended up with a kind of partial replacement of soybean. So yeah, I think it's something that would need to be obviously taken into account, but I guess it's still a pretty good performance, I think, overall, if we did, given that we did make the adjustments. So I think there's still quite a bit of potential. I know that the people who we would have worked with on the nutritional side in setting up this were very surprised to have the outcome that we did have, but then we've seen a similar outcome from Aarhus University and some of the other things. So it's not an outlier, but it probably does require some more trials as well to validate it further.
Yeah. Okay. Yeah. And then one more question, which did you also look at the legislation aspects? Does that play an issue here in any of the products that you produce, or is it not an issue because you use virgin grass?
Yeah, it's we didn't look at it in very much detail. I guess I don't know. I mean, whether it comes under the novel foods legislation or something like that, given that it's coming from grass, I don't really know. It's not something we looked into in much detail.
Okay. Yeah. Okay, thanks for your answer. There's one final remark, very interesting. Thanks for your answer. So that's a good response, I might say. So thanks again, James, for your presentation.
Thank you.
And then I would like to go to our last speaker of today, which is Marianne Volkmar. She studied biochemistry at the Technical University of Munich. And during her master degree, she turned towards bioprocess engineering. And since 2020, she is a PhD student at the Institute of Bioprocess Engineering at the Technical University of Kaiserslautern. And her research focuses on the valorization of so far unused materials and the fermentative production of base and fine chemicals. And today she will be speaking about pressed use of municipal grass clippings as medium for microbial fermentations. So I would like to give the floor to you, Marianne.
Well, thank you. Then I will share the screen. Is it working?
Yes, it's working.
Okay, perfect. Well then, thanks again for the kind introduction, and also welcome from my side. At our institute at the Technical University of Kaiserslautern at the chair of bioprocess engineering, we have quite a year-long experience of working with alternative and renewable resources. So here you can see some of completed and our current projects in this field. For example, we just finished a EU project focusing on the use of brewer spent grain, where we established a process chain where we used also solid state fermentation of the brewer spent grain, and also fermentations on the pressed juice, as well as fermentations on the further pre-treated pressed cake. We also had a project funded by the German government where silage was used for the production of organic acids, such as lactic acid, and also the production of ethanol. Currently, we have two projects funded by the German Ministry of Education and Research and also the Ministry of Nutrition and Agriculture. There is, on the one hand, the Green to Green project, where we use municipal green waste as a feedstock for microbial fermentations. Here, the challenge is that our feedstock is very diverse because the municipal green waste, it includes from grass clippings up to foliage and hedge and tree cuttings. It's a very broad variety. So there's a need for a very robust downstream process for the pre-treatment of the material and maybe also for the following fermentative processes. The second project we're working on right now is based on German ryegrass, so it's a more homogeneous feedstock, and this is a cooperation with growers to develop a cascade use of this special grass. As already mentioned, we are focusing on pressed juice from grass clippings. As we already heard today, the benefit from the pressing is that we have two separate fractions that we can both use. So we have the liquid fraction that we can immediately use for fermentations, and also we have the solid fraction, which we can also use after hydrolysis, which we can also use for fermentations. And as we already heard, the solid fraction is much easier to transport without the liquid part. Here at our institute, we're using two different pressing systems. So you can see a screw press. It's the same as we saw earlier, but in a much smaller scale and lab scale, and also a high pressure tincture press. So with the screw press, we have the rotating cylinders, which crush the material. And as you can see here, the resulting press cake is very fluffy. The tincture press applies a high amount of pressure, up to more than 400 bar. And as you can see here, the press cake is very compact and the grass structure is still intact. Due to the high pressure of the tincture press, we get a slightly higher yield of the pressed juice, but there's also less suspended matter in the pressed juice, so this might be beneficial if we need to filter the pressed juice afterwards. But the mechanical pre-treatment of the grass is the mechanical pre-treatment in the screw press is beneficial if we want to use the press cake, for example, in an enzymatic hydrolysis afterwards. The press juice we now produced contains valuable ingredients. For fermentations, the main focus lies on the carbohydrates, for example, the glucose, fructose, and cellulose or saccharose content, but also the protein content as a nitrogen source is of interest. The pressed juice also contains a lot of minerals, mainly potassium with up to 7 grams per liter, and chloride and sulfate with about 1 gram per liter, and also calcium. But as you can see here, the content varies widely depending on the location where the grass is growing, and the time of the year, and also the grass species. For example, you see here the composition of a mixed grass cutting in different time of the year, and you see that the content is, there's a really big difference in the composition. When we use the grass pressed juice for cultivations, it is necessary to sterilize it. And one way is to autoclave it. But as we have a carbon and a nitrogen source at the same time, there might be Maillard reactions that occur. So we lose quite a lot of sugars. As you can see here, the autoclaving process leads to a loss of about a quarter of the sugar content and about 70% of the protein content. So we need to consider this loss if we want to use it for the fermentation and compensate the loss of sugar in one way or the other. Or maybe we should choose another way of sterilizing, like for example, to sterile filter the pressed juice. On the next slides, I will present some results of using grass pressed juice for various fermentations with various different organisms. So here we cultivated Clostridium acetobotulicum, which is famous for the ABE fermentation. So it produces the solvents acetone, butanol, and ethanol. In the first diagram, you see the course of the cultivation in a standard medium under anaerobic conditions with 45 gram per liter of glucose. The glucose is metabolized into solvents, and we have an overall solvent yield of 0.24 grams of solvents per gram of sugar. When we tried to use pressed juice, autoclaved pressed juice, we discovered that the Clostridia are not growing at all. So that's probably due to toxic Maillard products. This is why we supplemented the media with sterile filter pressed juice. And this you can see on the second diagram. Here you can see that the glucose is also metabolized, and we have the production of the three main solvents, with a solid production for the solvents of 0.18 gram of solvents per gram sugar. Another example of a tested organism is Ustilago maidis. It's a fungus which is used for the production of lipids, and in our case for the production of itaconic acid. It is cultivated under aerobic conditions in the modified Debuchy medium with the composition shown below. Here we used between 50 and 70% of pressed juice, of autoclave pressed juice, and we supplemented the glucose concentration to a total concentration of 50 gram per liter. In the image, you can see the growth in the light microscopical image grown in 50% of pressed juice, and in the diagram to the right, you can see the growth in 70% of pressed juice. As you can see, the culture is growing exponentially, and the maximum optical density is reached after 4 days. Compared to cultivation in regular medium, the maximum OD is reached 1 to 2 days later, but the same maximum values are reached. But in contrast to a regular cultivation, where we achieved 14 grams per liter of itaconic acid, no itaconic acid was produced. So one possible explanation that we are examining right now is that it's due to the composition of the pressed juice, because the itaconic acid is produced under nitrogen limitations, and as the pressed juice does not only contain carbohydrates but also proteins, so as a nitrogen source, it might be that this is the reason that we don't see any production. So it is important to take into account the whole composition of the pressed juice and not only the sugar content. And it might be that the media needs to be adapted according to the organisms that we use. Another organism that we tested is Lactobacillus delbrueckii. It was cultivated in pure autoclave pressed juice. In the first diagram, you see the cultivation in a complex standard medium. Also, the composition is shown below, with carbohydrate content of 20 grams. Here we obtained a yield of 1.36 gram of lactic acid per gram of sugar. The higher than theoretically possible yield is described in literature and is because of the complex compounds we have in the medium. We cultivated the same organism in pure pressed juice of a mixed grass cutting. And here in the pure pressed juice, only with the addition of calcium carbonate for the control of the pH, we obtained 1.27 gram of lactic acid per gram of sugar. So already close to the comparison. And then we supplemented the pressed juice with media components of the standard medium to make up for maybe missing nutrients. And this way we obtained a yield of 1.58 grams of lactic acid per gram sugar. So this definitely confirms the suitability of grass pressed juice as a fermentation medium. We obtained similar results with the fourth organism I want to present, which is Saccharomyces cerevisiae. Here as well, you can see the fermentation in regular general yeast medium. In the first diagram, here we obtained a yield of 0.7 gram of ethanol per gram sugar. Below, there's the fermentation also in pure pressed juice, which is again supplemented with the general yeast medium components. And we obtain a comparable yield of 0.71 gram of ethanol per gram sugar. Without the supplement of the media component, we obtained 0.6 gram ethanol per gram sugar. So in conclusion, we can say that the grass juice is suitable for a variety of organisms, but it is important to take special requirements of the organisms into account and adapt the medium or the cultivation conditions accordingly. So we have a promising fermentation medium or fermentation medium supplement made from something that is so far mostly considered as waste. So here I would like to thank the BioBal Innovation Area and the German government for their support, as well as Professor Ulber and my colleagues and students in the lab, and of course you for your attention.
Thank you very much, Marianne. I would like to ask if there's any questions from the audience, because I didn't see anyone in the chat. Well, I have a question to start with, maybe on your previous slides, when you looked at Saccharomyces cerevisiae. Yeah, I saw that the glucose and fructose was not completely consumed in comparison to the top figure. Do you have any idea why that is?
That is correct. To be honest, I don't know what the reason is in this case. Okay,
and maybe a second question is, when you look at commercialization, did you think about that? So how do you think how promising is it to use the juice in this case for these fermentation purposes?
So far, that is still in the lab scale. So the biggest experiments we performed were in the 0.4 liter scale. So there's still a lot of upscale to do, of course.
Okay, so you're not in that stage yet.
Not yet. Yes.
Okay, thank you. Well, I don't see any other questions for you, so thank you again, Marianne, for your presentation. And then I do see a question from Gordon, possibly a question for James. How is the protein concentrate extracted from the press juice? I hope James is still here. Apparently not listening. So then since we concluded all presentations, now that there is some time for general questions or discussion, is there anyone who would like to bring up a question or a discussion point, yeah, for us to discuss, and maybe also for the audience?
Yeah, if the audience doesn't have, I do.
At Marcella, otherwise indeed I would have come up with a question for you or for any of the other speakers. So, well, if you have a point to bring up, please do.
No, I do have a general question on everybody using the grass juice. So of course, from Grassification, as I mentioned in one of my presentations, we were working with a frozen stream. So it was quite, everything already happened there. So there were several hours that it was exposed to weather, and then we froze it. So we killed, I think, everything that was already in there from bacteria, yeast. They didn't survive this freezing step. And also, I think it kind of changed, several physical properties. And then when we went to the grass juice algae operational group, now that we have the fresh grass juice being pressed, we do see that it has two phases. It has a liquid phase and a solid phase. And I was just wondering if this is something from our process, from the specific press that we are using, or if, James and Marianne, if you also see this. Or if you really get just a liquid that stays, homogeneous.
Yeah, it's indeed an interesting question, because we all use similar processes, but just a bit different. So did you by any chance have a look at that, Marianne?
We discovered the same thing. So we have the press juice, and usually we centrifuge it afterwards, because we still have this. We don't really know how to call it, this solid, the solid stuff in the press juice as well.
Okay, I'm happy to see that we agreed that we don't have a name for it. We call it slurry.
Yeah, something like that.
Okay, but then you see it as well.
Yes.
And do you see a difference between the two types of pressing that you are using?
In the tincture press, there's less of it, because there are in general less solid material in the press juice in general.
Okay, and then you're just working with the supernatant.
Yes.
Okay, no, that's interesting to know.
Did you ever look at the slurry part, as Marcella is calling it, or are you going to look at that? Or is that not interesting for you? Or did you analyze it? I'm also wondering if there's a difference in composition. Probably there is, and that also could open some new ideas on applying them.
We checked the sugar content, and we discovered that there are still carbohydrates in there. So now we are thinking about trying to use it as well for the fermentation. So try the fermentation without the centrifugation step. And see if those carbohydrates can also be used by the organisms.
Yeah, that's interesting. In the Grass2 algae, so we're working with farmers, and yeah, they say that they're interested. I think Chris is even here listening, so I hope I'm not saying anything wrong. But he was interested in testing the anaerobic digestion. If you have this stream, you just put it in the digester that's already working. If it boosts the production, great.
Okay, I do see a question popping up now, just out of curiosity. This pressed grass juice you are using has been filtered after extraction? Question mark.
We filtered it right after the pressing step.
After the pressing step, you you filter it.
Yes.
Yeah, and I guess that's looking at. Okay. Yeah.
Yeah, for us, we're looking into not filtering it because we're really trying to do something that a farmer would do, but we will compare and see how it happens. Yeah, how it goes, because as I mentioned, we do have a lot of organisms there. So yeah, as Mariana said, it might be necessary to filter and kind of sterilize it, but we'll see.
It would be good. So far, we don't have a really satisfying procedure to filter it because we didn't find a procedure yet to filter it in large scale, because even after 1 hour of centrifugation, it blocks all the filters almost immediately. So good luck with finding a possibility for filtering.
Thank you. I'll let you know if we do.
Thank you. Same here.
Sounds like a problem that occurs in many different media you work with. So I'm thinking also, Marcella, if you are going to look at that, we have another project, the WOW project, where we also had some issues, but then it was with bacteria. So I can look up what we came up with there, because there we also had these issues going on. Maybe a question for you, Marcella, as project leader of Grassification. Since we saw a lot of different applications this afternoon, which ones do you think are the most feasible ones in the end for commercial applications, or which will be the future? Do you have any comment on that yet?
That's so tricky. Yeah, I would say the applications that James presented are really already in a higher TRL level. Well, TRL is already level, so higher TRL. I don't know if this is the future, but I just think it's a good indication that you should be able to valorize this grass juice into actual products. From our side in Grassification, I have a tendency to agree with all that Mariana presented, that is fermentation. I would say it's such a rich stream in sugars and other organic molecules that my mind also goes to fermentation technologies to valorize it.
Yeah, okay. I see that Rosine still has a question about the solid components. Maybe, Rosine, you can ask the question yourself. Can you read the chat, Mariana or Marcella? So otherwise, I'm just repeating what's in the chat. It says.
Yeah, I'm just checking.
Yeah, sure.
So what is, I mean, that could explain the presence of solid components, that this grass juice is more like a suspension. Yes, I agree. It's more a suspension than a solution. So yeah, it can be indeed. I still didn't characterize it. I don't know if Mahina saw some protein, if there are proteins, but indeed I could think that maybe some So the pH is very low. So it can be that some proteins are just coagulating there, and that's why we also see a suspension. So it can be a little bit of fibers for sure, but yeah, the proteins can be coagulating and precipitating. So yeah, we still have to investigate more, but it's definitely a suspension. And I would say that we should take into account this slurry and yeah, part for valorization. Otherwise, it's really a huge part. So yeah, we cannot just look into the nutrients, the soluble part, and forget, pretend that it doesn't exist. Yeah, we have to think about something to do with it, even if it's just digesting it.
Yeah, okay, thanks. Any other discussion points anyone would like to bring up this afternoon? Well, I guess not. Yeah, well, we have still a couple of minutes left, but I think we had a long afternoon with a lot of nice, very nice presentations, and I'm very optimistic. Well, I already was, but even more about applications of the liquid of grass juice. To end this webinar, first of all, I would like to thank again all the speakers for their very interesting presentations. This afternoon it was taped, so it will be provided to all the people who are present later, and I think also the presentations will be shared as PDFs later to the people present this afternoon. And a final request, just in the chat pop up an evaluation form. So we would very much appreciate it if you would fill in the evaluation form so we can improve future webinars, which we are going to organize with Grassification. And then I can also already mention that somewhere at the end of this year, we will have the final event of Grassification, and you will definitely get an invitation for that. So you are very welcome to join there and hopefully live see some of the products that we have produced, both from juice and from the fibers of grass. So thank you all very much for being present today, and I hope to see you at some other event. So thanks.